Name a thing that people make. Fuel, electronics, medicine. This is the stuff of chemical industry, the stuff that makes up our lives. We want these things to work. We want them to be reliable. We want them to be affordable. We'd also like to avoid waste and minimize our impact. In sum, we want our chemistry to be both effective and efficient, but it's hard to overstate how difficult that is. The reality of lab work is that even with the best conditions, the best starting materials, we still have to invest significant time and energy to get a product that we could trust in something like a medicine. Contrast nature, which takes the burgers and fries you had for lunch and makes something as intricate as a hormone, a blood cell, a beating heart. How does that happen? Well, the secret is that nature does chemistry in a fundamentally different way. Instead of making your chemicals in a batch, your body has a distributed network of nanoscale protein structures that makes each molecule one at a time, exactly where you need it, perfectly, and orders of magnitude more quickly than you could in a lab. So researchers can't yet make something as complicated as a protein, but the question becomes, could something simpler work? To test this, we really try to model what the biologists think are the key features behind this efficiency. And one of these key features is that proteins are folded polymer chains. And they're so dense as to create a pocket where reactions can occur. This density is very important. So when we set out to fold the kinds of chains that we can make in a lab, the problem we run up against is that we can't yet replicate the density of a protein. My research question then is, instead of folding a linear chain like a protein, what if we chose a branched arrangement instead? The branched arrangement may not be the form of a protein, but it's something that we also see in things like starches and similar chemical structures. And the dense arrangement, or the branched arrangement, is naturally more dense. Now, little to nothing was known about the folding of branched chains, and when I set out to design the experiment comparing a linear and a branched chain, I realized that a common reaction could be used to grow a chain and connect it at the same time. This procedure is a lot more like knitting a structure than folding it, and it naturally results in products that can be twice the weight and a third less volume. It's my hope that this new class of nanoknit structures both challenges and complements the linear folding paradigm in my field, and ultimately helps people make their chemicals in a way that's as elegant as we can in nature.